identifying potential measures of stress during a captive to wild elephant reintegration.
Good morning. Today I'm going to be presenting on the process of investigating potential measures of stress during a captive to wild elephant reintegration that was facilitated by the Elephant Reintegration Trust in 2016. One may ask what the relevance is of measuring stress in elephants once they're in the wild. However, in certain systems, monitoring stress can be helpful in acting as an early warning system to highlight potential problems within an elephant population. Studies have shown reproductive and social status correlate significantly with stress hormone levels, for example. When looking at welfare in elephants, it stands to reason that higher welfare is achieved in more natural systems, where human intervention and management intensity is at its lowest. So we can expect that any given elephant population will sit somewhere on this welfare scale based on the specific local management strategies employed and the impact these have on elephants' natural behaviors. Resulting stress caused will change depending on the nature of the interventions, such as exposure to human infrastructure, including fences and mitigation strategies, immobilizations, wild captures, poaching, culls, and hunting, and ultimately, at the extreme, the confinement and forceful handling of elephants. Although some of these management interventions may be justifiable, if unchecked, they can become welfare issues. The Elephant Reintegration Trust is set out to advocate for improving elephant welfare. This is being done through facilitating the process of rewilding captive elephants, as well as through research projects focused on improving our understanding of this welfare management intensity relationship, which my colleague will be expanding on a bit later. I'll be sharing the findings from one of two reintegration projects that have already been undertaken since 2015. The study site where this reintegration occurred is a privately owned 10,000 hectare game reserve in the Waterberg region in Limpopo. The elephants were originally captured as orphans after culling programs that took place during the 1980s in Gonero National Park. They were trained and used for elephant-backed safaris in Zimbabwe till 2002, before they were translocated to South Africa, where these 6 individuals, 4 males and 2 females, were used in an elephant-backed safari operation for another several years until 2016. In that time, the herd has since increased to 12. The objective was to compare stress indicators as the reintegration process was carried out. Stress-related parameters were compared for the different stages at 4 temporal scales, going from broad to fine scale. These were grouped into captivity status, reintegration phase, looking at extreme and frequent disturbance events, and common immediate disturbances. On the broader scale, we looked at stress parameters while the elephants were in captivity and had any form of contact with their human handlers, compared with data collected after their release into the wild, which was on a scale of months. This captive phase period was further split into 4 phases. Phase 1, where elephants were still ridden by tourists as part of an elephant-backed safari operation. Phase 2, where elephants were not ridden, but still kept in individual stables at night and herded by human handlers during the day. Phase 3, where elephants were moved to an electrically fenced 100 meter by 100 meter open-air boma, where they were closed in together at night, but allowed to roam free within a 5-kilometer radius of the boma during the day. And Phase 4, where the gates of the boma was left open and the elephants were allowed to roam freely on the reserve. And then lastly, ADAPT refers to the final phase, documented 5 weeks after their release, where the elephants were roaming freely on the reserve, and this was on a weekly scale. Then throughout the entire study period, note was taken of days during which the elephants were exposed to potential extreme and frequent disturbance events on a daily scale. This included activities such as the fitting of tracking collars, physical treatment of wounds, injection of the bull with GnRH, herding of the elephants from the stables to the new boma, and aggressive physical interactions with other species such as rhinos were also noted. At the finer scale, during each observation session of the group of elephants, any immediate potential disturbances present in the environment were noted continuously by the minute. These common immediate disturbances documented included number of vehicles visible within 100 meters of the elephants, other visible or audible human disturbances in the area, and then any natural disturbances caused by weather or other species were recorded. In order to monitor progress during this reintegration, 3 stress parameters were selected to try to represent physiological and behavioral stress responses at each of the stages. These stress parameters included monitoring stress levels through testing dung samples for fecal glucocorticoid metabolite levels, monitoring of temporal gland secretion status, as well as close monitoring of behavior changes through focus on established stress-related behaviors and stereotype behaviors. Cortisol levels can be measured non-invasively through dung sample analysis. As this method is quantifying cortisol levels that have been excreted in the digestive system, there is a variable lag period that has to be taken into account, which can make it hard to link increases in stress levels to actual events or causes, but it is still an effective method to monitor relative stress levels in an individual over time. And then temporal gland secretion status was scored from 0 to 4 based on the presence or extent of the secretion. This is an instantly visible physiological response triggered by high excitement events. So stress levels from dung are a nice way to standardize stress levels across a time period and between individuals, while temporal gland secretion measurements can be visualized immediately and note made of the triggers. To demonstrate this instantaneous response, this is a video of an elderly elephant cow moving behind her herd. She starts out with no secretion visible, but then within a few seconds we see she suddenly starts streaming from her temporal gland. This is likely in response to the pressure and discomfort of this game viewer that persists at following behind her. Here's just the same clip again, but zoomed in to be able to see how, in a matter of seconds, she goes from no secretion visible to streaming in response to this trigger. Lastly, we assessed stress through quantifying certain body language behaviors for individuals over the different periods as an indicator of the extent to which they were experiencing stress. Key stress-related behaviors that were noted in the study included behaviors such as walking warily with the tail up, head held high and ears out, head shaking, twitching and twirling of trunk, sudden pause while listening, bundling together in a defensive fashion with heads facing out, various fleeing behaviors, and vocalizations such as screaming, roaring, moaning, and growling were also noted. And then as the extreme, any display of stereotypic behaviors were noted. These are behaviors generally associated with captive elephants that display frustration in these ways, sometimes due to a lack of stimulation from their environment. In elephants, this culminates in rocking or swaying the body from side to side at a consistent pace, as seen in this clip, and sometimes as head bobbing, where elephants can be seen consistently bouncing their head up and down. Okay, having a look at our findings, in all the graphs, the shaded bars represent findings for the group, while lines and markers represent the findings for each individual that make up this group. At the broader scale, just comparing captive versus free status, we see that stereotypic behaviors completely subsided. So that swaying behavior we saw on the previous slide from that female in the group was never seen again in this whole group following the release. Then physiologically, the stress levels when comparing captive to free elephants were very similar and showed no significant difference. And when looking at temporal gland secretion extent, this did increase as the elephants transition from captive to free. If we then hone in on a breakdown of the reintegration phases, looking at the frequency of certain behaviors, it is clear that during the release phase, stress-related behaviors did significantly increase, but then decreased after a period of 5 weeks. A similar trend was demonstrated when looking at the extent of temporal gland secretions observed. Cortisol levels appeared to show a lag response, where in the first year, stress levels went up, but by year 2 and 3, the levels were almost at the same level as when they were still being ridden. The consistently high stress parameters observed during the release phase may seem unexpected. However, we must remember this is the first period in these elephants' lives where they were free to make decisions around foraging and movements themselves, decisions they've never been completely free to make, pointing to possible insecurities where they're essentially needing to build up their confidence with decision-making in response to their environment. At the third temporal scale, we see that the extent of temporal gland secretion was significantly higher for the elephant study group during days in which extreme disturbance events occurred. As the temporal gland secretion mechanism is somewhat likened to crying in humans, these occurrences are strongly related to emotion, but are not necessarily always negative, especially during something like this release, where they would have been experiencing lots of stimuli, which could have been overwhelming. Not to mention these elephants were very habituated to people, hence possible initial insecurities when these handlers disengaged and they were left to make their own decisions. The extent of stress-related behaviors during group observations were significantly lower in the presence of common disturbances, while the extent of temporal gland secretions was not significantly different for the elephants during sessions when common disturbances were present in the immediate environment compared to when not. When taking into account responses of individual elephants' age and sex, there were significant differences noted between individual elephants for cortisol levels, as seen in the top graph, with one subadult cow standing out in particular with significantly higher cortisol levels compared to most of the other elephants. Significant differences were also noted between individuals for the extent of temporal gland secretions, as illustrated in the bottom graph, with one adult cow also standing out with significantly higher temporal gland secretion occurrences recorded. This is consistent with findings from other studies that adults, and females even more so, secrete more than younger individuals. But this being said, we see how there are still individuals that can stand out more than expected, highlighting a need to take into account individual differences when it comes to studying behavioral responses of such socially complex animals. So why does this all matter? When evaluating the stress incurred by elephants through a reintegration process, results showed that certain stress-related parameters increased during the initial release compared to when in captivity, but this did eventually return to pre-reintegration levels. Then positively, we saw the stereotypic behaviors were only recorded during the captive phase, but immediately ceased after release. And these findings also indicate that fecal glucocorticoid metabolite levels, the extent of temporal gland secretion, and stress-related behaviors all proved effective in explaining the changes in stress experienced by elephants when being reintegrated from captivity into the wild. Interestingly, we saw that the habituation of captive elephants to humans alters expected responses to stimuli that are classically defined as disturbances in a wild elephant. And moreover, we can finally do more than just pay this process lip service, as the study showed that successful reintegration of elephants that have been raised in, and in this case, spent over 20 years in captivity, is possible without compromising welfare in the context of stress. A huge thank you goes to the Reserve for supporting this research, the incredible handlers that facilitated this reintegration, and the EMS Foundation for funding and supporting this research and its presentation at this conference. Thank you.
Thank you, Tammy. We have time for one quick question.
Hi, thanks, Tammy. I was just wondering, have you compared the data to translocations of wild elephants to see, in either breakouts or moving a herd from one location to another, to see how stress levels change following a new environment for wild elephants?
So we haven't. I'm trying to think if we've documented that. Obviously, the short term, it has been well studied. Those short term effects of those big changes is definitely a spike in stress, which tends to level out after a few weeks. But obviously, being integrated into new social systems is something we would want to add into this study to be able to compare on a similar scale. Yeah.
Much.